Hello everyone. Let's study cofactors today. We have studied in the previous lectures that most of the enzymes are proteins. Now, this protein part of the enzyme in many cases cannot alone function to catalyze the reaction. It requires help of a non-protein part, that is cofactor, so that this entire complex of the main enzyme protein part and the non-protein cofactor together will form a functional enzyme, and this functional part, the functional enzyme, will catalyze the chemical reaction. So, what we can say is the main protein part of the enzyme, the core enzyme, is the apoenzyme. This is not functional on its own. Okay, it cannot function. It cannot change the substrate into product. It requires binding of the cofactor. So the main protein part is the apoenzyme. Then we have the non-protein part, which is cofactor. And together, the apoenzyme plus cofactor will form the entire functional enzyme, which is holoenzyme. Holoenzyme is apoenzyme plus cofactor, and this holoenzyme is functional enzyme. It will be able to catalyze the specific chemical reactions. Now, let's see what are the different types of cofactors. Now, we can divide cofactors into two types, organic and inorganic. Organic cofactors can be divided into two types. First, there are some organic cofactors which are tightly bound to the apoenzyme. Apoenzyme is the protein part, right? So, to this main core enzyme part, protein part, there are some organic compounds which are tightly, permanently associated. Such type of cofactor is called a prosthetic group. And opposite to this, we have some organic compounds which are loosely or transiently, that is temporarily bound to the apoenzyme. These are called coenzymes. Okay, very important point. The tightly bound cofactors, organic cofactors, are prosthetic groups, and the loosely bound or transiently bound cofactors to the apoenzyme are called coenzymes, right? Both prosthetic group and coenzymes are organic in nature. Now, let's see some important examples. Prosthetic group. Okay, first. Heme is the prosthetic group for enzymes peroxidase and catalase. Both these enzymes are important for breakdown of hydrogen peroxide. Now, for both these enzymes to act, these enzymes require the heme group attached to it. Heme is the prosthetic group for peroxidase and catalase enzymes. This heme is part of active site of the enzyme. Very important point. Next, we have the coenzymes. Examples, we have NAD and NADP. Both are actually derived from a vitamin which is niacin, the B3 vitamin. Okay. Here NAD stands for nicotinamide adenine dinucleotide. What is NAD? Nicotinamide adenine dinucleotide, and if you attach a phosphate group, you will get NADP. Both are crucial for specific enzymes activity. Like you will study about NAD in cellular respiration, and you will study about NADP in photosynthesis. Okay, so NAD acts as hydrogen acceptor in many steps of cellular respiration. And this NAD or NADP, or even there are more like FAD, flavin adenine dinucleotide, these are required as coenzymes for specific enzymes to work. So far what we have seen is cofactors are of two types, organic and inorganic. Organic ones are of two types, tightly bound and loosely bound. Tightly bound ones are prosthetic group, like heme is the prosthetic group for peroxidase and catalase. Next, loosely or transiently bound cofactors are coenzymes, and examples of coenzymes are NAD and NADP, and both contain vitamin niacin. Now let's come to the inorganic one. Inorganic cofactors will obviously include the metal ions. There are so many enzymes, rather many of which you all have studied in photosynthesis and respiration also, which require some specific metal ion for its activity. Without these cofactors, enzyme cannot do any function, like the enzyme activity is lost if cofactors are absent. Example, we have zinc as a cofactor for carboxypeptidase enzyme. Carboxypeptidase is a protein digesting, that means proteolytic enzyme, and for this enzyme to act, it needs to be associated with zinc. Zinc is the cofactor here, and zinc actually connects both to the main enzyme, apoenzyme part, and also to the substrate with the help of coordination bonds. So remember this, coordination bonds are very important so that these metal ions like zinc can form by association with the enzyme part and the substrate part. Okay. So these are all the examples of cofactors which you will have to study for your NEET examination. Now it's time for the knowledge drill. Try to answer questions in the comment section. First, name the cofactor for carboxypeptidase, and second, name the proteinaceous part of the enzyme to which the cofactor binds. I hope this session was helpful for you. Thank you.